Hi, welcome to the second part of this module. What we learned in the first part is what exactly immunotherapy means, right, and then we have seen how we can extract T cells from blood or from spleen. When we talk about spleen, right, we have to take spleen as you have seen in the video, right, we are taking spleen and then we are loading the spleen in a media, and then the T cell isolation process is that you take the spleen, crush it, take the splenocytes, from splenocytes you can extract or isolate T cells using T cells isolation kit. The kits are two types, one is positive selection kit, one is negative selection kit. Then these are your naive cells. So, you have to activate those cells and that activation can be done using Dynabeads. Now, once you have these T cells, what you will do with this T cell? You have to see what is the concentration, what is the ratio of CD4 to CD8 in the given number of cells that we obtain from spleen or we extract it from the blood, right. Now, this will be your T cells without interaction or without interacting with tumor microenvironment. Then we have seen how the static process is done. Take a trans well, at the bottom of the well you can load the spheroids, matrigel, load the well, trans well, load the T cells and let the interaction occur between T cells and spheroid. There will be some releasing of chemokines, because of which there will be change in CD4 CD8 concentration. Now, you treat those same cells with an immunomodulator or immunotherapy drug and you block one of the immune checkpoint on T cells. In this case we are talking about PD-1 and see the interaction. Right. So, now you will be able to see the change in CD4 CD8 ratio when you talk about control, that is cells, T cells which are not treated with any immunomodulator and T cells which are treated with a particular immunomodulator. So, we know whether the immunotherapy drug is working or not. We can understand the efficacy or we can evaluate the efficacy of immunotherapy drug. So, then where is the problem? The problem is that this system that we are using or that is currently being used is static system, and like we mentioned in last module that our body is not static, it is dynamic. Of course, I am sitting here without doing anything, it is static, but still the body within it is dynamic, right. Blood is flowing, heart is beating, right, blood is flowing. So, when a patient has a cancer and we give a particular drug will dissolve in the blood circulates in the body, and that is how the drug diffusion to a particular site would happen, right. It is not that when you give a drug will go to a particular site and stick there for 24 hours. So, can we mimic this in vivo situation, that is within the body, onto a platform that is in vitro and see the effect of immunomodulators on T cell? That is our question, that is our interest. And is there a difference when we do that? So, same experiment, take out T cells. Now, from where you can get spheroids, from where you can get this tumor, you can get the cells from the patient, grow the spheroids in the lab using 96 well plate. We will talk about this 96 well plate cell culture in one of the modules, one of the lectures. So, you take the cells from the patient, from the site which is tumor, extract the cells, then grow spheroids in the laboratory. We have seen how spheroid looks like. You load this spheroid in a microfluidic chip, float T cells in the microfluidic chip next to the spheroids and see the interaction and see the CD4 CD8 concentration. Now, you treat this T cell with immunotherapy drug, again flow it in the microfluidic chip, have some interaction and see the change in concentration of CD4 CD8 cells. By doing so, are we getting a difference in terms of result between a static experiments and dynamic experiments? If yes, then we should go for dynamic experiments rather than static. Another point, about 68% of patient responses well for the immunotherapy drug, around 32% are not responding well. So, why that 32% patients are not responding for a given therapy? Because it is already FDA approved drugs, or these drugs that are given are already approved by a particular drug governing agency, right, approval agency. It is all within the limits, within the rules, with lot of results that you need to show that the particular drug is effective, lot of clinical trials that you undergo. So, finally, the drug comes to the market. Then when a patient is taking the drug, why drug is not effective? So, can we design a platform that is patient centric? That is our quest, right. So, if you see the screen, what we have seen? We have seen immunocheckpoints. We have seen if we block one of the checkpoint on T cell, what will happen, right. We are assuming that CD4 CD8 concentration will be different. We have seen how the T cells are activated from the resting state in case of in vivo, and how dynamics could activate the T cell from its resting state. We have seen this is called transwell, right. Now, let us see. Now, you can read, research on cancer immune interaction has accelerated recently because of the enormous potential of immunotherapies, including blockade of immunocheckpoints, right. Blockade. We can block immunocheckpoints in unleashing potent anti-tumor immune response. Blocking of programmed cell death protein PD1. PD1 is on T cells, right, see on T cells. So, blocking program cell death protein on T cells using monoclonal antibody has been successful in boosting anti-tumor immunity, you see here. So, current immunotherapy studies using in vivo models do not replicate the physiological relevance, as they inherently contain non-human host cell. So, the present studies that we go are in mice, like animal models are in mice. Now, the results that we get, we cannot say this is exactly replicating the human, since the animal cells are the host cells, right. Now, meanwhile, in vitro 2D studies, like we have seen, right, transwell, do not replicate the actual tumor microenvironment and may provide inaccurate data regarding the response of cancer cells to immunotherapeutics. You see, one thing is PD1, blocking PD1 can be effective. Second thing is in vivo models, animal models will not replicate actual tumor environment because of the animal host cells or non-human host cells. Third thing is 2D in vitro model do not replicate the actual tumor microenvironment, and thus it is possible that they may provide inaccurate data regarding the response of cancer cells to immunotherapeutics. Okay. So, what to do now? Static experiment. Let us see how static experiments are done. We have just discussed. Let us see here. To study the interaction between cancer cells and T cells in static condition, transwell plates, you see transwell plates, 0.4 micron pore size, 12 millimeter diameter from Corning Life Sciences can be used or will be used, right. Cancer cells will be cultured in 12 well plate. It can be cultured in 12 well plate, where T cells can be cultured on a semi permeable membrane inserts placed on each cell. The plates are incubated at 37 degree, 5% CO2 atmosphere, right. In the experiments involving immunomodulator, the immunomodulator will be added to the T cells into the insert. That is what we have said. What we have said, that you take this well, load the T cells, right, and everywhere there is matrigel, there is a tumor spheroids here at the bottom and T cells are here with media, right. You have another well, you load T cells, you have spheroids, here again have T cells, right. Wait for 24 hours and see CD4 CD8 ratio, right. Here wait for 24 hours, see CD4 CD8 ratio. The difference between this one and this one is, in this case the T cells are treated with immunotherapy drug, right. This is control, just T cells. After 24 hours we see CD4 CD8 concentration. T cells treated with immunotherapy drug, after 24 hours we see CD4 CD8 concentration, right, and we compare. This is what it is written here, right. And of course, when you do this, you have to keep the plate in an incubator to maintain the 5% CO2 and 37 degrees centigrade atmosphere, so that the cells would not die, and the T cells can be kept in a immunocult, which is the media for keeping the T cells alive. So, if this is the method that is used and the results are obtained, there are some key questions that arises. First question is, why immunotherapy is not effective for all the cancer patients, right. Is it because the studies are done in static, in vitro platform or in vivo mice models where the host cells are animal cells? And can we develop an in vitro patient centric dynamic platform in which we can load the spheroids with matrigel and study the effect of immunotherapy drug on T cells by passing T cells next to the spheroids within the matrigel? right. So, that is how we had to create, that is why we had to create a dynamic platform, that is a microfluidic chip based study we will perform to understand the difference between static results and dynamic results. So, at present there are no microfluidic platforms, that is in vitro, for studying the effect of immunomodulators on CD4 CD8 T cells. Thus, there is an urgent need to design and develop an engineered microfluidic platform replicating 3D tumor microenvironment to evaluate the effect of blocking immunocheckpoints on CD4 CD8 T cells in a dynamic environment, right. So, if you want to check effect of immunomodulators on CD4 CD8 T cells, we need a dynamic platform, but currently there are no microfluidic platforms that are used for this kind of study. So, if the effect of immunotherapy drug using static platform on, of course, CD4 CD8 T cell ratio is different than dynamic platform, if the effect of drugs on CD4 CD8 in static platform, if it is different than dynamic platform, it is very interesting to study further. For that, you can see we are proposing to develop a microfluidic system for studying the effect of immunomodulator on CD4 CD8 T cells. And what is this platform? Let us see. There is a inlet port, right. You can see there is a microfluidic chip that goes here and merges here, this goes from here, goes all the way, it merges here, this is the outlet, this is inlet, this is outlet. In here there is a circle where you can load the cancer spheroid. In center you can load, in this region you can load cancer spheroids. You can see here spheroids are loaded here, right, seeding spheroids for matrigel. This is outlet for T cells, inlet for T cells, these are channels. The surface is from PDMS, the bottom is glass, and this is the interaction area where you can see here like this area, right, where you are loading the spheroid, right, spheroid is here with matrigel. So, this area is where the interaction between T cells and spheroids would happen. There will be releasing of chemokines, right, and change in CD4 CD8 concentration would occur, right. So, this kind of chip can be potentially used to study the effect of immunomodulators on T cell, right. Now, like I said, we always need to perform a simulation before we actually fabricate a device. Okay. So, before you go for any fabrication, we have to always go for simulation. In microfluidic chip, you need to perform analysis such as velocity, stress in the device, whether the flow is smooth or not, right. So, if you see in the screen, you can see the simulation results when we are flowing a drug through the microfluidic chip, and you can see that the shear stress plot of the microfluidic device is found to be less than 1.1 into 10 to the power minus 10 Pascal, and is uniform across the length. And we have not shown you the shear plot, but it was found that shear plot is the value is this, while the velocity plot which is shown here, the material field region in static and hence is shown as 0, this is at 0 because it is at static, right, while the velocity of T cells near the interaction area is sufficiently low to realistically mimic the in vivo condition. Velocity increases to 0.7 micron per second near the inlet and outlet regions due to tapered geometry. So, here the velocity increases little bit, however, in this particular area the velocity is sufficiently low to mimic the actual flow inside the body. The flow within the from the capillaries to the tumor is close to 0.35 micron per second or less than 1 micron per second. So, realistically this is mimicking the in vivo situation that happens in the body. So, we can use this particular device and study further what is the effect of immunomodulator. So, let us see. This is the process flow. This is the process flow. Since this is a process flow, we need to understand how these things are done, and this is one step process, okay, very easy, one step, single step process. So, you see here A is SU82075 will be spin coated on a silicon wafer at 1000 rpm to form 150 micron film. So, if you see this one, this is SU8 spin coated on silicon wafer, right, and we have about 150 micron thick SU8 film. Second is microchannel will be formed using UV lithography, this is B. C is SU8 will be developed and cured at 150 degree, so you can see it is cured. D is PDMS will be poured on SU8 mold and cured at 120 degree for 15 minutes. E would be PDMS will be detached from SU8 mold. F would be microchannels will be imprinted in the SU8 in the PDMS, and holes for inlet and outlet will be created. Finally, you can see here PDMS will be bonded to glass using oxygen plasma bonding and couplers will be attached to realize the device. Easy, very easy, right? Let us see. Let us see this thing how we can fabricate the device. So, let us see here. First point was you take the silicon wafer. Second point was you spin coat SU8 on silicon. Third point is you load mask on SU8, right, and SU8 is which kind of photoresist? It is negative photoresist. Okay. So, after SU8 you coat SU8, you have to do soft bake and then load mask, right, and you need to form structure which will help to create you channels, right. Okay, this is your mask, it can be dark field. Now you expose your wafer with UV. After exposing, next step is, after exposing, the next step would be to go for hard bake. SU8 is little bit different. So, you have to perform hard bake before developing SU8. Okay? Hard bake. Now, soft bake is generally done at 65 degrees centigrade, hard bake is done at 95 degrees centigrade. The time depends on the thickness of SU8 material. It is already given in the manufacturer protocol. Okay. So, after soft bake and you load the mask and you expose the wafer, then you have to perform hard bake, followed by developing in SU8 developer. When you do that, what you get? You get SU8 like this. Why? Because negative photoresist. The area which is exposed will become stronger, area which is not exposed will become weaker, right. Now, what you will do with this? On this SU8, you pour PDMS. And you cure PDMS at 120 degrees centigrade for 15 minutes. After that, you peel off the PDMS. So, when I peel it off, it will look like this, right. Correct. Now, I take a glass and this PDMS and this glass, I will perform oxygen plasma, I will perform plasma, right, around 100 watts and about 10 millitorr pressure. Once I perform oxygen plasma, I will bond glass and PDMS. So, this would be my channel, right, and this would be a microfluidic chip. This would be a microfluidic chip, right. So, I had to use only one mask process, you see, only one time we have used mask, single mask process. So, now if you want to see, you can see here once again what you are looking at. You are looking at SU8, right, spin coated on silicon. Then we perform photolithography to form channels on silicon, to form the pattern of SU8 on silicon. Then we cure the SU8, then we after curing the SU8 we get the mold, which is right over here. This is our mold, right. After mold we pour PDMS, on the mold. After pouring PDMS, we have to cure PDMS for at 120 degrees centigrade for about 15 minutes. Following that, we will peel off or we will detach PDMS from SU8 mold, which you can see in E. After detaching, you have to insert holes in this PDMS, right, in this particular 4 section through holes. Right. So, holes for inlet, outlet and for loading material with spheroid will be created, and finally, this is PDMS. So, we will perform oxygen plasma bonding. Once oxygen plasma bonding is done, we will connect the couplers, we will attach the couplers, right. So, if you see here the device fabrication, this is in detailed device fabrication process. And as we have discussed, the microfluidic chip will be fabricated using photolithography and PDMS glass bonding. A 4-inch silicon wafer will be used as substrate, which is here. B and C, the mold will be prepared using SU8. SU8 2075 will be spin coated at 1000 rpm for 1 minute on top of glass wafer with metal electrodes to obtain uniform layer of SU8. The SU8 will be pre-baked at 65 degree for 7 minutes followed by 90 degree. This is not metal electrodes, we do not require metal electrodes here. SU8 will be pre-baked at 65 degree. Like I said, pre-baking is at 65 degree. Now, this for 150 micron of SU8 we require 7 minutes of pre-baking time, followed by 90 degree for 25 minutes. After pre-baking, the SU8 will be exposed to UV lithography for 25 seconds. Post exposure bake will be performed at 95 degree centigrade for 15 minutes before immersing the wafer in microchem SU8 developer, right. So, once we do that, we can obtain channels with 15 millimeter length, 1 millimeter width, chamber size of 2 millimeter. Chamber size is what? So, let us draw it once again, so you understand in detail. We have something like this. Okay. And here we have inlet, here we have outlet, and here we can load spheroids. Okay. So, T cells, let us say T cells are flown from here and they are take out from here. There is a peristatic pump. So, T cell goes inside and flows back here, right. It is a circulation from here to peristatic pump. Now, T cells will flow in this area like this and in this area like this. Alright guys, it is very easy, and then it will interact. You see, T cells can diffuse through this region, if there are chemoattractants, and after 24 hours of flow we will measure CD4 CD8 ratio. That is our idea. Okay. So, for this kind of work we have now fabricated a microfluidic chip, right, microfluidic chip. So, slits that we are talking about, slits is this area, right, where the spheroids are there and the T cells will flow. These are the slits, right, both sides there are slits. The slits will be 0.1 millimeter, about 100 microns. For interaction between T cells and cancer spheroids in 3D gel, the width of channel will be kept close to upper range, which is about 600 microns of the pulmonary vessel diameter, right. So, the channel is about 600 micron. PDMS elastomer ratio is 1:10, we know it. After covering PDMS, it will be peeled off. To realize the device, PDMS will be bonded to glass, right, and of course before that we have to create the holes using acupunch, right, you have to create holes using acupunch. So, once the device is ready, then what you will do? Once the device is ready, we have to perform experiments like we were discussing. So, you can see here in detail what we are doing. So, there is a tumor, right, there are T cells in the tumor, right, there are T cells on the in the vessels flowing next to the tumor, there is a extracellular matrix surrounding the tumor, right. Similar situation we have to mimic on our microfluidic chip. So, you can see here very clearly, right, this is the area, center area where we can load spheroid. These are the channels, right, through which the T cells will flow. So, now what we are doing? We have to so this particular situation we are trying to mimic in this microfluidic platform, and in the center of the channel there is a matrigel with tumor spheroid which will work as this particular section, and the channels through which the blood will flow, right. Here we are flowing T cells with media, right. So, we are trying to mimic this particular blood vessel by creating the channels. Okay. So, this is again the same device, there is a glass at the bottom, there is a PDMS, there is an outlet port for T cells, there are inlet port for seeding matrigel and spheroid, there is an inlet port for T cells, channels are there and interaction area between T cells and spheroid. So, what does figure shows? The A shows tumor microenvironment interaction of tumor with T cells, B shows microfluidic platform replicating a tumor microenvironment, C will show 41 mouse based cancer cell spheroids embedded into the extracellular matrix. PDL1 is shown in only few cells to avoid ambiguity in the diagram. D will show after flowing T cells without immunomodulator here for 24 hours in neighboring channels to spheroids. PD1 binds to PDL1 suppressing CD8 expression, while adding immunomodulator to T cells is shown in E representative diagram, and it shows that adding immunomodulator to T cells circulating it for 24 hours next to cancer spheroids causes increase in CD8 T cells, right. And finally, the F, this figure shows the dimension of the interaction area, region for loading spheroids and embedded in matrigel and channels. So, this is what the diagram shows, right. Now, if we have this particular device with us mimicking the in vivo situation onto in vitro platform, what you will do? What you will do? You will perform experiments. So, you can see here the channel area A shows the bright field image of T cells, you can see few T cells here, right. B bright field image of matrigel, this is matrigel without any T cells. C shows bright field image of interaction area here after circulating T cells. D shows bright field image here of interaction after T cells, circulating T cells with anti-PD1 next to melanoma. So, what we see is that the T cells enters the melanoma if you use anti-PD1, right. You block the immunocheckpoint on T cells will cause T cells to go nearby tumor and start killing the tumor. Finally, what we see E is fluorescence microscope image of DAPI and actin stain melanoma spheroid infiltrated by CFSE stain which is T cells with anti-PD1 after circulating on the microfluidic device, right. You can see T cells here infiltrating the tumor microenvironment, and some of the live videos that you will be able to see on YouTube of T cells infiltrating tumor, you will be able to see the T cell starts eating the cancer cells, literally eating the cancer cells. So, if we see the results, we can clearly see that when we have control that the T cells that is activated at when it is taken out from naive to activated cells at T equals to 0, we have CD8 to CD4 concentration as 30 to 38. When we have static with spheroid at T equals to 24, the CD8 concentration to CD4 concentration is 17.7 to 63.3. When we have control at 24 hours not doing anything, we have 35.3 and 35.4, which is similar to this, right, not too much difference. So, its control is okay. And when we have dynamic with spheroid dynamic experiment, the CD8 concentration is different than CD4 concentration, you can clearly see here. Thus, we can see that compared to control either at 0 or at 24, the results for static and dynamic is statistically significant, while if you just consider dynamic experiment with static, right, then also you can clearly see that the results are statistically significant. That means that we get different results from static platform compared to the results that we get from dynamic platform, and that is why we should go for dynamic platform for understanding the immunotherapy or evaluating the efficacy of immunotherapy rather than using the static platform which is used right now, right. So, these are all flow cytometry studies, flow cytometry scatter plots comparison of percentage of CD8 T cells with respect to measure before and after exposing T cells to melanoma spheroids in static and dynamic condition for 24 hours. So, thus what we understand is that the results obtained from static experiments are different than the results obtained from dynamic experiments. I am not claiming that static is better than dynamic or we should go for dynamic, but now what we can say is that if you get the cells from the patient, right, then using these cells we can grow this kind of spheroid, right. Using the cells we can grow a spheroid which will look like this. So, take the cancer cell from the patient, grow spheroid in the laboratory. Now, you can extract T cells from the blood of the patient, right. Then take this T cells and this spheroid, load into this device, load the spheroid here, right, and then take your T cells, take the patient T cells and extract and flow the T cells next to spheroid using peristaltic pump and using a microfluidic chip, right. Then after 48 hours or 24 hours you can just of course, it depends on you have to optimize the time, just see CD4 CD8 concentration, right. Then take the same T cells and treat the T cells with a particular drug, let us say anti-PD1, flow the T cells again next to the spheroids for 24 to 48 hours and again check the CD4 CD8 concentration. Then if the drug is effective, you will see the CD4 CD8 concentration very different when compared to when the drug is not effective for that particular patient. Thus, we can use this device as a patient centric platform, since we can try to evaluate the different available drugs for particular patient, and from the results that we obtain, let us we should get some idea that which drug to give to a patient, or from given number of drugs a particular drug is more effective for a particular patient. Thus, this device can act as a patient centric device after we get lot of results, right. So, what we learn in these modules, what we learn here is what is immunotherapy, right, how T cells looks like, how can T cells be isolated from the spleen of the mice, right. This is how T cell looks like, this is a 20x image. So, that is why you can see it bigger. How we can form spheroids, and what is static way of doing the experiments, right. How can we activate the T cells, then what are the key questions that one has to address when we are looking at immunotherapy. Now, immunotherapy is extremely vast, we are just addressing a very small section of immunotherapy, alright. What kind of microfluidic chip we can design, what kind of simulations we can do, right. We can do shear stress, we can do velocity, we have shown you the velocity plot here, I have shown you velocity plot in this particular module. How we can fabricate this device using one mask step, single mask step. How we can design the channel, what are the process flow for device fabrication, right, and then we have seen how the microfluidic chip can be utilized and for understanding the effect of anti-PD-1, that is our immunomodulator, right. So, just go through it. It is extremely interesting and burning topic in the area of research, particularly for those who are interested in applying their engineering knowledge, particularly microengineering knowledge, microtechnology or MAMS, whatever you want to talk about it. The photolithography that we have learned, now you see we can utilize it here. Now, when you have a mold and you keep on getting the design, it can be considered as a soft lithography, since you can create lot of channels by just pouring PDMS and curing PDMS on the mold, that is your SU8 mold, and curing the curing the PDMS and just stripping it off. So, you get a channel in PDMS, right. So, you can get lot of PDMS, lot of channels in PDMS, a lot of PDMS based microfluidic chips using a single mold, and that is why this can be considered as a soft lithography, right. We are not really performing a photolithography, mask and everything once we have the mold. So, this is a beauty that we can create lot of devices when we go through this particular route, right. I hope you understand the importance of immunotherapy. I hope you understand how you can use microfluidic chip to understand or to evaluate the efficacy of given therapies, particularly we are talking about immunotherapy drugs, right. Lot of things to learn. So, I always believe that if a teacher gives you 10%, a student has to put 90% emphasis to learn more. And if you are stuck somewhere, feel free to ask me. I will respond to your question to the best of my understanding, and let us create something new. Let us create novel technologies that can be translated to clinics, and hopefully we can save few patients' life by understanding this particular technology and implementing it and designing a new kind of devices. So, I hope you will go through it once again. Not so easy to understand, that is why I have tried my best to keep it as simple as possible, right. However, if you are confused, feel free to ask me through the beautiful NPTEL forum that you guys have, and I will respond to your question. Thank you, take care, I will see you in the next class.